We were discussing about the cultivation, collection, and processing of crude drugs. In that, till now we have discussed about the pest and pest management. So today we are going to discuss that one of the factor affecting that is the growth regulator, right? So these are nothing but plant growth regulator (PGR). So coming to the PGR, it is a hormone like synthetic organic compound, right? Like humans are having hormones, no, like that plants also having the hormone. These hormones are helping them to growth, cell division, cell multiplication, internode division, right? Like that different growth it will support them. So these are like synthetic organic compound, right? But these are not nutrients. Other than nutrients, generally what is the role of nutrients or food? It will help them for the normal growth, but here hormones are giving them extra support in a small amount. It modifies the growth and development either by promoting or inhibiting the growth. Sometimes it is also inhibiting, not only promoting always, right? Sometimes if the growth it is more, so we can use certain types of regulators which will inhibit the growth or control the growth of the plant at different stage, right? Coming to the general plant hormones, the phytohormones are broadly grouped under five major classes, namely auxin, which participate into the cell elongation. Second one, gibberellin. These are participating or functioning onto the cell elongation plus cell division, like translate into the growth, right? Cell division also they will be helpful. Third one is the cytokinins. These are cell division and inhibits the senescence, right? This also. Then fourth one is the abscisic acid. Abscisic, the abscission of leaves and fruits. Then fifth one and last one is the ethylene. Ethylene, they are participating into the promote substance senescence and the epinesty and fruit ripening. The ripening of the fruit also, these are helpful in the different plants. Coming to the first one, auxin. Indole acetic acid is the best example of auxin. Here the structure also given 3 indole acetic acid. You can see the carboxylic acid, it is third position, and nitrogen, tertiary nitrogen group, it is there in the first position, right? So auxin increases the plasticity of plant cell wall, right? And is involved in stem elongation. The production of auxin occurs at shoot tips and a developing seed, right? This area of the plant, this production of the auxin hormone, it will be there, and the main role it is that to create the plasticity of the plant cell wall. Okay, physiological effect of auxin. The first one is the cell elongation and expansion. It is also having the role into the cell elongation and the expansion of the plant. Initiation of adventitious roots. Root will start their growth, initiate their growth, right? Abscissions, loss of leaves. It is also playing the role into the loss of leaves. Flower initiation, fruit development, apical dominance, and lateral branch growth are inhibited near the shoot apex, but less so further from the tip. So these are the role. You can see here evidence for the role of auxin in apical dominance. You can see here high auxin if it is there, the shoot tip auxin source here. I told you that the production of auxin will be there in the shoot tips. So this is the shoot here tip. On the tip you can see high auxin concentration will be there and inhibit the axillary bud. The axillary buds, these are the axillary buds, so where the near the leaves you can see that some eye like structure, some curve like structure it is there, that is the axillary buds. It is going to inhibit whenever the auxin high concentration will be there into the plant. Whereas low auxin concentration, the concentration will be low at the bottom side, where the it will play as a role in the developing auxiliary buds. Here the auxiliary buds, how it is developing, you can see here. Compared to this auxiliary buds, it is more developed, where the concentration is low when the particular place. In this picture we can see that shoot tip removed. If a tip here, the tip was there, no, if it is removed, the auxiliary bud no longer inhibited. If this side it will be cutted, they will start growing from this side, from the bud side, auxiliary bud side, they will start growing as a plant, and the branches will develop. So this is the difference where the concentration, if it is increasing, how it is inhibiting the auxiliary buds, and the concentration is low, it is growing the auxiliary buds. Wherever you are cutting the root shoot tips on the top, they will start growing from their different buds where the even the concentration is high. With synthetic auxin and without synthetic auxin, you can see the auxin also can be synthesized. If synthetic auxin, if it is there, the adventitious roots growing from the stem tissue, these are nothing but stem only, right? So you can see here, if the synthetic auxin it has given to the particular plant stem, we can see the root, yeah, from the stem root growth we can see. Where auxiliary, the auxin, synthetic auxin, it is not there in the particular sample for the same plant, here no root growth can be seen, right? So that is the difference. If you are giving the auxin, we will be able to develop the root even from the stem, and if you are not giving auxin, no root growth can be seen. That's what the importance of auxin we can understand with this picture. Coming to the next, auxin, like growth regulator in synthetic mode, indole butyric acid, IBA. Then second one, 2,4-dichlorophenoxyacetic acid, 24D also you can write in abbreviate. And the third one example, 2,4,5 trichlorophenoxyacetic acid. So these are the three example of auxin which can be used. Second one is the gibberellin or gibberellic acid. It is also known as GA, both in capital, right? So this is the second plant growth regulator. You can see here in the structure, the structural formula of gibberellic acid. Here carboxylic acid as a functional group into the this five-membered ring, right? So here the hydroxyl group it is attached here in both the side, and if you practice, you can, the methyl group it is here. If you practice this one, you can make this, right? Here one benzene ring, the five-membered ring, and one benzene ring, it is attached like this in double bond, right? So this is not that much difficult. So what is the role? Let us understand. Produce in roots and young leaves. These are the location in the plant where gibberellic acid will be there and produced or biosynthesized. Now known to be essential for stem elongation. Now after the so many research, it is it has been cleared that the essential for the stem elongation, whenever you have required a stem elongation, it is useful there. The physiological effects of gibberellin. First, cell division. Of course, the dividation of the cell wherever required into the plant, especially into the root and leaves, this hormone is helpful. Cell elongation. It is also elongating the cells, different plant cells, and also stimulate the seed germination. Whenever seed we are showing into the soil, right, and providing all the requirement like moisture contain moisture zone and proper soil, it will initiate or encourage the seed germination, and seed will start germinating, and it is also stimulate the flowering. The whenever the plants are not flowering for the long time, we can give this gibberellin, and we can see the into the plant that flowering it is initiating, and it is also stimulate the fruit development. Whenever sometimes in some plant fruit, they will bear the fruit, but at the same time they will not uh, reach to the maturity level, or they will not develop. So in that case we can stimulate by giving this one. the commercial use of gibberellic acid, you can see without gibberellic acid and with gibberellic acid. How the in grapes we can see the larger fruits that are easier to clean are attractive in markets. So whenever you are going to purchase grapes in the market, what kind of grapes you are attracting? You're attracting towards the big one, sweet one, yellow color, or the the bunches of the grapes should be very much proper, so that you can think that this is the healthy and tasty, and you are purchasing those kinds of grapes. If it is like that, means you are rejecting this, because if you are not using gibberellic acid, without gibberellic acid, or the plant is not able to produce the sufficient quantity of gibberellic acid, the fruits are going to be like this. And if you are using gibberellic acid, the fruits bears, or they will be stimulated themselves, and they will bear in good size. Third one, cytokinin. These are easier natural, the either natural or synthetic. The natural example is Zeatin, and the synthetic example is Kinetin, right? Compounds with the significant growth regulating activity. You can see Zeatin example here, the structure, how the for five member ring and six member ring is attached together with the different nitrogen atoms, right? Wherever one or more than one nitrogen atom it is there in the basic skeletal, it will be considered as alkaloids. Here you can see the how in the side chain it is going, and the alcoholic functional group it is there, right? CH2OH, and here methyl group attached with the carbon atom, and by fulfilling the valency of carbons, how they have attached. So you can practice this structure to draw when giving the example of cytokinin. Let us discuss about the physiological effects of cytokinin. The first one, the promotion of cell division. Of course, it is helpful into the promotion of cell division. What we have seen in the last two hormone, here also the same role we can see in the cytokinin. The second physiological effects, it is external application of cytokinin promotes the growth of lateral buds, right? The buds which will becomes later, and those buds we can see the growth if you are adding this one. Cytokinin breaks the dormancy of many seeds and also promotes the germinations. Seed dormancy we can break by using cytokinin, and also it will promote the germination, seed germination, right? The fourth physiological effects, that is application of cytokinin delays the process of aging in plants, right? Plant aging can be delayed by using cytokinin hormone. Here in picture you can see that treated with cytokinin one plant, and without treating with the cytokinin another plant. So in treated with cytokinin, the plants, the leaves you can see how they are green and healthy, and even in the bottom side also, and in the shoot side also, the axillary part also you can see how much they are growing. Whereas untreated plant, it is the leaves are dried, either dried or whitish yellow color, and they are about to fall down from the stem, like that we can see here. So you can understand the importance of cytokinin by seeing this image. Cytokinin delay the leaf senescence. You can see that in the image. This is cytokinin with the same age of the leaf. How this side of leaf, these four leaves, you can see here, they have the leaf has become dried, and without chlorophyll. Here you can see the chlorophylls in between the leaves. Even though these are also dried, but you can see the chlorophyll here in the plant. These two plant example. This is treated with cytokinin. This is without treated with cytokinin. So leaf senescences can be delay, or it will not be earlier. The next hormone, that is the abscisic acid. Abscisic acid is produced chiefly or mainly in mature green leaves and in fruits, right? So green leaves you can see into the plant and fruit where this will be producing, and produced in mature leaves, especially under stress. Roots then transported to shoots. Once it will be produced, then it will be transported by xylem phloem and to the shoots parts also. So you can see the structure here, the carboxylic acid, the functional group here, and how this is the methyl group. It is attached, the hydroxyl group, right? With double bond, the two methyl group attached here into the six-membered ring. So this is also not that difficult. Okay, double bond. Don't forget, whenever you are drawing this structure, these two double bonds has to be there by to fulfill the valency of carbon, right? So this also you can practice while drawing the abscisic acid structure. Let us discuss the physiological effects of abscisic acid. The first one is stress response. Stimulate stomatal closure, inhibit premature germination of seeds, right? uh, then induce bud dormancy in a variety of plants, and seed dormancy maintenance also it will be done by the abscisic acid, right? So here you can see the stomata. They have given the god cells and the cell, the stoma opening, whereas the stoma closed, right? The transportation of potassium and water also it will be done in the mechanism of opening and closing of the stomata, right? Cell turgid, right? So here you can see that stomata how it is opening. That's what we have discussed into the here. Stimulate the stomata closer. So whenever you are adding abscisic acid or ABA, it will stimulate the closing of stomata. So here we have used the abscisic acid. So we can see the how stomatas are closing, whereas it was opened without abscisic acid. But whenever you are adding abscisic acid, these are closed, or these are stimulating the closures of the stomatal thing. The last one, ethylene. Ethylene is a simple gaseous hormone. It is usually present in minute quantity. It is synthesized in large amounts by tissue undergoing aging. You can see the structure. It is very easy. Two carbon by fulfilling this valency 4 bond, and hydrogen atoms it is attached in both the carbon 2 2, right? So it is very easy to draw. So whenever writing your answer, you just try to draw all the structure of the hormone. If you practice, you will be able to do that. Coming to the physiological effects of ethylene, fruit ripening. So the first important, very very important, that is the ethylene. It is useful into the fruit ripening, the ripening of fruits. Whenever you can see into the mango or in the orange or in other fruits, if they are fruiting but at the same time they are not able to ripe, because ultimately mango, it is you can sell or you can consume that in the ripening stage only, and unripe fruit also it is being used, but most of the marketing it is depending on to the ripe fruit. So ethylene hormone we can use for the ripening of the fruits. The second effect, that is the leaf and flower senescence, leaf and fruit abscissions, that controlled separation of plant part from the main body. Flowering, ethylene inhibits flowering in most species but promote it in a few plants such as pineapple, bromeliads, and mango, right? So generally its role is to inhibit the flowering, but except these three plant or three fruit, the pineapple and the bromeliad and mango, these fruit three it is not inhibiting the flowering, right? You can see in the carica papaya or papaya fruit 3 day after storage at room temperature, and the papaya fruit exposed to ethylene gas. 100 pp and 100 ppm, and 3 days after storage at room temperature. These fruits also we are storing at the room temperature for 3 days, and these fruits also we are storing at the room temperature for 3 days. So when 3 days we are storing with the ethylene gas, because as I told you, the ethylene, it is gaseous hormone, so it is in the gas form generally. So if you will use those gas, it will help for the ripening of the fruit. So those who are doing for the cultivation of this fruit in the commercial purpose or for the marketing purpose, they are using ethylene gas, and the papaya fruit will become nicely ripe, which can be attractive and also easy to eat. The next example, banana. Ethylene in fruit ripening. You can see in the diagram 1, it is completely unripe, whereas in diagram 2, little bit the yellow is ripening started because you started using the ethylene gas. In third one, the maximum parts have become yellow. In fourth one, only few parts having green color. The fifth one, only top and the down green, remaining yellow. Here in sixth one, complete ripe, complete yellow. And the seventh one, it is excess ripened. We can say that's what black spots has been developed onto the surface of the peel of the banana, right? So that's what the more use of the ethylene also.